Hi, everyone. Welcome to our poster presentation session. Welcome. We were originally going to do this in the format of breakout rooms, but we've made a last-minute call to instead keep everybody together. And so we're going to ask our presenters to present once in order, and they will do a 1 to 2 minute presentation of their research and slide as they were going to do. And then we will have a few minutes for questions if you have any questions for them. And then we'll move to the next person and so on and so forth. And if a question comes to you at the end, by all means, write it down. We may have a few extra minutes at the end for a few extra questions. So I'm going to ask, I'm going to keep admitting people, but I'm going to ask Atefe if she would like to present first. And the rest of us, we can turn our cameras off while she's presenting and then join again afterwards.
Okay. You guys see my slide?
Yeah.
Yeah.
Can you make it a little bit bigger? Yeah,
Yeah.
there you go. Perfect.
Okay. Today I'm going to talk about actually a part of my PhD project, which is focused on haptoglobin and its contribution to the symptom severity in ME and long COVID. But specifically, I'm going to talk about the role of haptoglobin in long COVID and the symptom severity in long COVID. You know that you might know that the previous work from our lab showed the contribution of haptoglobin phenotypes to PEM severity and cognitive dysfunction in ME, as HP21 correlated to the more severe symptoms, and HP11 emerged as a protective variant. Considering the noticeable clinical overlap between ME and long COVID, we were aimed to investigate the potential contribution of haptoglobin to cognitive functionality in long COVID. So in this study, we performed HP phenotyping in individuals who experienced their first COVID-19 infection between 2020 and 2022, which was confirmed by public health. And our patient group reported an average symptom duration of 8 months without hospitalization, and were compared with individuals who fully recovered. All the participants underwent a PEM inducing a stress challenge, which is a messenger device that's applied to the arm of the participants and induces the pulse type pressure to their arms. And all the participants completed 3 study visits over a 6-month period at 3 months intervals. And at each visit and also at each time point before and after the stress test, they completed health questionnaires and engaged in cognitive evaluation using brain check test. The results showed that in those with HP21 and HP22 phenotype showed a decline in brain check performance at 90 minutes after the stress test versus T0, T90 versus T0, as you see at the bottom of the slide in the figure A and B. While those with HP11 phenotype showed a slight improvement, suggesting greater cognitive resilience in this specific group of patients. And also across the 3 study visits, participants with HP11 phenotype showed a significant improvement in cognitive performance, while those with HP21 and HP22 phenotype remained consistently lower and showed no meaningful change, as you see in tables A and B at the right side of the slide. And it suggested greater cognitive also resilience in HP11 over time also. So in summary, HP11 and HP22 phenotypes are contributing to cognitive impairment in long COVID, and also HP11 is associated with cognitive resilience in long COVID. And thanks for your attention. And it's the end of my presentation.
Thank you so much.
Of course.
We'll ask if anybody has any questions. If you do, you can raise your hand and we will be able to open your mic, or you're welcome to type it in the chat if you have any questions. And if there aren't any, then we'll go to the next speaker, and there's always an opportunity at the end, maybe if there's a few questions. But this is great. It's so lovely to see you guys presenting your research. Okay, no questions, so we'll move to Braden, and thank you so much, Atefe.
Of course.
Am I correct in assuming that you guys can see my screen?
Yes, we can.
Perfect. One of the kind of greatest misunderstandings, I think, with ME/CFS is this idea of patients exhibiting a lot of deconditioning as an explanation for their symptoms. So what we've gone to try and quantify in the last year or so is to what extent some of these symptoms might be explainable by deconditioning. And so what we've done here is we've taken a cohort from a previous bed rest study that was conducted by the European Space Agency, where healthy people prior to COVID underwent 60 days of head down tilt bed rest, so absolutely no movement for 60 days. And we also had a cohort that involved long COVID patients, as well as ME/CFS patients who were diagnosed prior to the COVID pandemic. And both the long COVID and ME patients exhibit post-exertional malaise. So technically, the long COVID patients, in most circumstances, qualify as ME patients. Perhaps unsurprisingly, what we find is that in both bed rest or deconditioning, we see this decrease in VO2 max aerobic capacity, as well as in our patient groups. And this is also reflected in our gas exchange threshold, which is our non-invasive marker for lactate accumulation. However, if we look at the point at which the lactate accumulation occurs relative to their maximal aerobic capacity, we see that actually in the ME/CFS patients, this occurs earlier than we would expect in not just healthy controls, but also compared to deconditioning, suggesting that they have this not just absolute earlier onset, but an earlier onset relative to their max. As well, we have this VO2 heart rate response throughout an exercise test, which is reflective of either stroke volume or oxygen extraction at maximal exercise. And if anybody's familiar with David Systrom's work, they've shown with invasive CPETs that there's indications of ME/CFS patients having poor oxygen extraction. And this is somewhat what we see here in the breath-by-breath measures. However, we can't necessarily conclude that because it's measured at the mouth rather than invasively. So overall, the whole body exercises seem to be completely different from what we'd see typically in classic deconditioning, where we don't see this impaired oxygen extraction. Further, one of the biggest hallmarks of any bed rest study is this severe decrease in muscle fiber cross-sectional area or muscle atrophy. And this is something that we don't see with either the long COVID or the ME patients. As well as in deconditioning, they typically lose muscle mass prior to any kind of changes in capillarization. We actually see the opposite here with the ME patients, where we see this loss of capillarization preferentially compared to any loss of muscle mass. And this is also related to their fiber cross-sectional area, but we have a lower capillarization in the ME patients for any given muscle mass, suggesting that that there's not an adequate supply for any nutrients or oxygen. One of the biggest predictors for exercise capacity or just whole body metabolism is also our mitochondrial function. And what we can see here are two different measures, our oxidative phosphorylation capacity and our SDH activity, which is a marker for mitochondrial density. And so what we typically see in deconditioning is a decrease in the oxidative phosphorylation capacity. However, this is primarily driven by a loss in mitochondria, whereas in the ME patients, the loss that we see in the oxidative phosphorylation capacity is more attributed to a decrease in mitochondrial function. So overall, what we tend to see are, even though at the superficial level of just whole body exercise, ME patients might resemble deconditioning. However, the underlying alterations are completely different. And I'd like to thank you all for your attention and be happy to take any questions now.
Thank you so much. This is a lovely presentation. And we heard a little bit yesterday from Dr. Wust about some of this as well. He mentioned you a few times during his presentation. We have a question from Pietro. Pietro, do you want to unmute?
Yes. Hello. I'm a patient expert, and I would like to know if do you finding point to a single practical biomarkers in this study, or small panel that could be turned into a simple clinical test to detect the alteration and maybe support a diagnosis of ME/CFS or long COVID?
Yeah, so in these particular measures, we don't have any specific biomarkers. They are more corroborations of what Dr. Systrom's already seen in the invasive CPETs with the oxygen extraction. We have other data and other studies ongoing that are more related to the biomarkers themselves. This is more of a mechanistic study. I hope that answered the question.
Yeah, thank you.
Fantastic. We have a question in the chat about would light activity help augment the number of mitochondria?
Oh, that is a very good question. This is actually something that we've been thinking about ourselves as to, yeah. So the idea behind this being exercise of any capacity does stimulate mitochondrial biogenesis. And I think what we tend to see kind of reflected in our long COVID patients versus the ME/CFS group is that small amount of additional activity and training status, like recently, might augment any losses of mitochondria, but then over time with a subsequent deconditioning. So ME patients being sick for longer, they have more time to lose certain things. So this is seen in both the capillarization and the mitochondrial densities. Then whether some light exercise is enough to completely augment that, we're still not sure. And then the question is, what kind of exercise would then be considered light? So it's the balance between PEM exacerbations and stimulus. So very good question, and unfortunately, I don't have a very good answer for it.
I think that was a really good answer, and I think the challenge always is with a question like that is you're balancing multiple things. We might be able to help something over here and hurt something over here, and so which one is more important, and how do we test it safely? So these are excellent questions, and you gave an excellent answer. PEM being what it is, and our understanding of it still being unclear, and patients knowing sort of where their limits are. It will always be a question of what's light, and for somebody it will be very heavy for somebody else, and also what can we do without causing PEM safely.
Exactly, yes. Yeah.
But your presentation's excellent. I'm going to leave it there for now, and if anybody has any additional questions, again at the end we may have a few minutes, but great job.
Thank you.
So up next is Corinne.
Yes, I will share now. Yes, can you see it all right and hear me?
Yes, that's perfect.
Okay, so just to let you know, I will present in English, mais s'il y a des questions en français, je peux tout à fait répondre en français également. So hi, I'm Corinne. I'm a PhD student in Alain Moreau's lab. I'm mainly working on ME and specifically the cognitive related symptoms in ME. And here I'm going to present a comparison that I did between ME patients, long COVID patients, and healthy controls of their cognitive impairment symptoms. So as my colleague Atefe explained, we're inducing PEM in participants with our little maneuver here. It looks like a pressure measuring device, but it's much softer. It lasts 90 minutes. And we took we did cognitive assessment with the participants right before and right after the PEM induction. And we use a tool called Brain Check. It's a little test. It takes around 10 minutes, and it's different cognitive tasks, assessing mainly attention, mental flexibility, digit symbol substitution, and Stroop or cognitive execution. I forgot the name. The cognitive function, executive function. There we go, got it. And the last two are for memory. We had 3 different groups. So as I said, healthy controls that recovered 2 weeks after COVID infection. Long COVID participants, we still had symptoms 6 months after their first infection, and ME participants who had different triggers for their disease, and most of them had a ongoing disease for quite longer than those 6 months in long COVID. For the COVID, we wanted a confirmed diagnosis by public health and no hospitalization whatsoever. Here we assessed the severity of symptoms using typical questionnaires, and it was highly significant when we compared both ME and long COVID to healthy controls as expected. So on the right-hand side, you see the results, and each panel is for each test done with Brain Check. And within each panel, you see T0, which is baseline, and T90, which is immediately after the PEM induction. So the first thing we see looking like broadly at the figures is that long COVID patients and ME patients all really react to our PEM induction because their performance significantly worse than healthy controls, or SC for short COVID, in almost all of our tasks. So that's very interesting, and it also shows that our little PEM induction works quite well for these people, so we can capture real changes. Then both long COVID participants and ME participants have cognitive impairment, especially after PEM induction, as expected. But we see that there's no real big difference between the two. So it's really hard to use that kind of assessment to differentiate between the two conditions. And what we see also is that long COVID seem to experience a little the cognitive impairment that's a bit stronger than ME participants, as we can see in the different panels. For example, where my cursor is, most participants are mainly in the lower part of the graph, so it's below the 50th percentile, and it's more extended for the ME participants. This is attention, but we see it for almost all of the tests. Mental flexibility, here we see something similar. The same for almost all of our different tests. Don't have a clear explanation for this yet. Several studies have shown that when you are just diagnosed with the disease, or within the first few years, it's a bit harder to adapt, and sometimes your symptoms are a bit stronger. So that could be one of the explanations. I'm gonna dig a little bit further into that for the rest of my project, and I'll be taking questions if you have any. Thank you.
Excellent. I would have one question, I guess. What happens when, how will I ask this? We know PEM is usually delayed by 1 to 3 days. And so are you guys taking measurements during that time to be able to capture PEM?
Not in our long COVID cohort. What we do though is 5 days post-PEM induction, we have a questionnaire asking the severity and the frequency of 13 different symptoms. And we explain clearly that it's whether these symptoms are caused by the induction and not general symptoms that the participant may feel every day. So that's a way of capturing maybe, okay, that person had more insomnia, for example, or that person had more muscle aches than usual. So that's something we can also see, and it will be, and because it's 5 days post-test, it can be developed later on than right after it. For our ME cohort, we are doing also another brain check especially for these participants, not for all the cohort, but these 87, they did another brain check 5 days also post-test, which I have analyzed those results. It's mixed results. Some people get better, others are still I wouldn't say maybe strong, but are still experiencing PEM from what we see. So yes, so we also have that 5 days post induction questionnaire for these participants, and we really see sort of different profiles. Some that are reacting stronger, some not that much. It's very varied.
Okay. I guess my suggestion as a patient would be to move measuring for PEM to day 2 or 3 to capture as much as you can, because by the time day 5 comes, some people may, if they're milder, actually may be starting to get a bit better. So that that day 2, 3 is sort of that sweet spot where you're probably going to find the best, loudest signal of PEM.
Yes. Actually, we do. It's just that it's partly analyzed on my side yet, but for our ME cohort still, because they're doing a bit longer protocol, we have a journal, so they can write down the symptoms and what they are feeling and everything. So we do have that information on the everyday post induction. I have started analyzing it, and what we see is, yes, indeed, on the same day of the test, the day after, and the one after, usually the symptoms are a bit stronger. It's not quantitative result as they are with questionnaire, but it's still very interesting to see. And I'm now looking into whether what symptoms are really put forward by the patients, what they're feeling more, and to what extent. So that's what I'm trying to work on also on the side.
Excellent. Somebody said, I was a participant in that research. Would it be correct to say if my situation has worsened, the results can now be different?
I'm not sure I understand.
Yeah, I'm not sure I understand either. Masha, do you want to, you can either unmute and ask the question, or just type a little bit more and we can go back to it. I'll go to Pietro and I'll give you a moment to type what you'd like to type. Pietro, do you want to ask a question? Did I see you?
Oui, alors, je vais en profiter pour poser la question en français à Corinne, qui pourra peut-être répondre en anglais. Mais je me demandais si dans le test qui a été fait pour provoquer le malaise post-effort, on s'assurait que ça provoquait bien un malaise post-effort chez la personne en question, ou on partait du principe que ça en déclenchait un, en fait. Parce que, par exemple, dans mon cas, il m'en faut plus que ça pour aller loin dans les troubles cognitifs ou dans le brain fog, quoi.
Ouais. Je vais répondre en français, je vais peut-être traduire après, juste comme ça j'ai la question en tête. Je dirais que nous, on part de la prémisse que ça va causer un malaise post-effort, mais on le voit dans les différentes mesures que l'on fait, qu'effectivement, chez certaines personnes, ça semble pas nécessairement en déclencher un. Par exemple, là, je parle du brain check parce que c'est particulièrement là-dessus que moi je travaille, mais il y en a effectivement que au temps 0 puis au temps T90, il n'y a pas vraiment de différence. Fait effectivement, peut-être que c'est pas suffisant pour déclencher quelque chose parce que c'est quand même assez léger, en effet. Ce qui est intéressant, c'est qu'on peut, avec justement les journaux remplis par les participants, puis le test 5 jours plus tard, on peut voir si est-ce qu'en fait c'est que ce malaise après effort-là se déclenche tout simplement plus tard, ou comme vous dites, c'était peut-être pas suffisant pour le déclencher. On a assez de... je dirais qu'on ramasse assez d'informations, tant cliniques que biologiques, et avec les questionnaires pour être capable de dresser un portrait relativement clair. Mais c'est sûr qu'effectivement, il y a certaines personnes qui semblent peut-être pas trop réagir, si on veut.
Ok, merci.
So what I would caution is that you're definitely going to see some things happening in the short term after the provocation, but PEM may not be seen until days 1, 2, 3 for most ME patients, and the long COVID patients with PEM, we see the same thing. And so there's lots that happens immediately, and then there's lots that happens as part of that PEM process. So it's just being able to differentiate which of those two things may be happening. So all the data is important data, but it's whether or not you're seeing PEM that, and I've had that conversation with Dr. Moreau too, and then I think, we see that with the CPET. If they do in research, if they use a 1-day CPET, they rarely get the information that they need to get because they don't have time to witness and trigger that PEM effect. So that's why 2-day CPETs are so important in research. Chantal has a question, which is in French, and if I read it, my brain's going to burn, so I might let you
Je
read
peux
it.
y aller. Oui, oui, yes, of course.
And then we'll move to the next presenter, just so we have time for everybody to present.
Sorry to interrupt. I just wanted to say I strongly agree with Sabrina. Sabrina there. She makes some excellent points, and just to add that in as someone else agreeing. So
Thank you so much, Michael.
Sabrina, I don't see the question, so maybe I could either send it to me and I'll answer directly or something.
I can translate. The question is there a stratification amongst the levels of severity? I think, Chantal, if I got that right.
Ouais, should I answer in French or in English?
Answer in French for Chantal, and then
OK.
you can explain.
Dans les résultats que je présente actuellement, non, j'ai pas séparé, juste parce que je suis pas encore rendue là dans mon analyse. Pour mes patients EM, mais mes patients, les participants EM, j'ai commencé à faire cette analyse-là, oui, puis on voit qu'il y a une différence de sévérité. So, short answer in English is that in what I'm presenting right now, no, I didn't stratify according to severity. In my main project, which is for now focusing mainly on ME participants, I did that stratification, and yes, we see an important difference.
Perfect. And I'm just going to go back to Masha, and then I'm going to move on. But Masha said, I'm trying to unmute as a participant in that research. Is it safe to say that 2 years later, if my symptoms have worsened, that if I was due to do the test again, would the results of the test be different?
That is
Probably, but I don't know if you can guess that really, because we have so many fluctuations in ME that it's possible that if the symptoms were worse, that they would show up on your test. Yes?
Exactly. It's really, I mean,
You'd
have to do it again, I guess.
Yeah, we wouldn't know for sure, Masha, unless you did the test again. But of course, there's a chance based on the test that they're doing that it would show. Okay, everybody, I love all these questions. This is great, but we have a few more presenters. So Corinne, maybe we'll come back to you at the end if we have some time. And our next presenter, Gazelle, is ready.
Yes. Let me share.
I just love the engagement too. And remember, like after each presenter, if you have questions, we'll have a few minutes. So by all means, type them into the box and I'll do my best to ask them, or you can unmute when the time comes.
Okay. Can you see my screen?
Yep.
Okay.
Perfect.
Hi, everyone. My name is Fazal Azimi, and today I'm pleased to present, to present a part of our recent study that is titled Uncovering the Dual Role of FGF21 in Myalgic Encephalomyelitis and Fibromyalgia Pathogenesis. As all of us know, ME and FM are complex overlapping diseases that characterized by fatigue, pain, PEM, and also cognitive dysfunction. Despite the decades of research and many efforts, their mechanism remain unclear, and there's a lack of reliable biomarker for differentiating this disease. On the other hand, FGF21 or fibroblast growth factor 21 is a key regulator that influences energy metabolism, mitochondrial activity, and cellular stress adaptations. Given the evidence of metabolic dysfunction and mitochondrial stress in ME and FM, we hypothesize that abnormal FGF21 signaling might contribute to disease severity and symptom diversity for patients. So the objective of our study would be to investigate how stratification by plasma FGF21 levels reveal clinical and cognitive heterogeneity in ME and FM and also ME plus FM patients. So for the methodology of our study, we measured the levels of FGF21 in using ELISA in patients with ME, ME plus FM, and FM patients, and also healthy controls. All the participants were divided into 3 different groups based on FGF21 levels in low, normal, and high FGF21. That's the low groups, the levels of FGF21 is below 15, and also for the normal, based on the literature, was 51 until 200, and also the high group that is above 200. So clinical and cognitive features were assessed using standards and validated questionnaires and brain check tests that my colleague Corinne referred to brain check briefly, that these tools covering fatigue, PEM, and all the symptoms related to these comorbidities. On the blue table, you see that the demographic distributions of patients in terms of mean of age and sex, that they are in the comparable range that across different groups. So for the results of our study, our results revealed interestingly distinct FGF21 related patterns between 3 groups. For low FGF21 levels, this group, low FGF21, were linked to worsened PEM perceptions in FM groups, also increased PEM severity and immune autonomic symptoms in ME groups, and also poorer mental health in ME plus FM patients. On the other hand, high FGF21 levels correlated with better cognitions in ME, but also greater physical fatigue in ME plus FM patients. So all these data suggest that FGF21 levels reflect distinct pathophysiological subtypes. So we can conclude that FGF21 acts as a disease-modifying factor instead of just defining a simple biomarker for this disease. And interestingly, several medications are defined and known to enhance the FGF21 activity, like metformin, that can help for therapeutic target for a special subset of patients, for example, with lower FGF21, that we can alleviate the symptoms of pain for them. Also, on the other side, there are some medications and tools that can decrease the activity and the intensity of FGF21 to also balance the function of this protein or hormone. So these data show the importance of metabolic precision medicine that targets patient subtypes based on molecular signatures and profiles, instead of just defining a special method, homogeneous method for all groups of patients. And thank you for your attention.
Fantastic. Any questions? Remember, you have 2 options. You can unmute or you can type it in the chat.
Okay.
Do you have any questions for this presentation?
Can we just leave the poster up while the questions are happening?
Could you put the poster back up?
Yeah, sure. Let me just a few minutes. Okay, again, I share. Okay.
Perfect.
There you go.
Alicia, do you have a question?
Not right now. I need to look at it more.
Okay.
Yeah, sure. And also, the part of our results are published in a well-known article that I put the QR code for your curiosity.
Fantastic. I saw lots of emojis. I think people have learned how to use the emojis at the bottom and you got lots of emojis. So.
Yeah.
Okay, so if there are no questions right now, we will move to the next presentation. And again, at the end, we might have a few minutes for extra questions. So please feel free if you have any to write them down and we'll come back to you.
Okay. Thank you for your attention.
Thank you for presenting. And we have Yasima next.
Yeah, hello.
Hi there, welcome.
Thank you. Is that fine for you?
There it is. Yep, perfect. Thank you.
Okay, perfect. Hello again. My name is Yasama Vahdani, and as a PhD candidate in Dr. Alan Moreau's lab, today I'm going to present part of my recent result under the title of Molecular Insight into a key regulator of autonomic dysfunction in myalgic encephalomyelitis, or ME. As you probably know, we have several symptoms for ME based on Canadian consensus criteria, and one of the main important symptoms would be dysautonomia, which is divided to different parts such as orthostatic intolerance or OI and postural orthostatic tachycardia syndrome or POTS, which is a condition when you change your position from sitting or lying to standing up, you may feel some increase in your heartbeat. And since ME and POTS share some similar clinical feature and symptoms, as you can see in figure number 1, interestingly more about 25% of ME patients develop POTS, like tachycardia, increase or decrease in their blood pressure. It was interesting to find out what are the exact causes for both comorbidities and how we can distinguish between them. Previous research from our lab focused on discovering more about microRNAs or small non-coding RNAs as a link between genetic and environmental factor, because they are somehow a modulator of the regulation of the gene expression. So after a pan-epigenomic study, I found out that there are 2 interesting microRNAs significantly expressed in ME patients with POTS syndromes. And after going through literature like target scan to analyze the binding site of these microRNAs and related genes, IPA, which is Ingenuity Pathway Analysis software, in order to see the predicted and probable gene microRNA interaction or network. And also the most important one was patients demonstrating higher score in the self-reported questionnaires, specifically about POTS symptoms like irregular heartbeat, which was question number 51 in DSQ questionnaires. We found out that one of these MERS, which was MER 181A5P, is both clinically and experimentally relevant to excessive heartbeat related to patients with ME plus POTS. Also, target scans showed several and multiple binding sites with these microRNAs with 3 important calcium handling genes such as N61, ATP2A2, and ATP2B1, which made me curious to also validate the functionality of this microRNA in a relevant cellular model to see the potential effect. So as you can see in methodology part, in order to do that, we differentiated the human iPSC into mature cardiomyocyte and transfected them with different lentivirus treatment in 4 different treatments of lenti-MER, which overexpress the microRNA in cells, lenti-off-MER, which inhibit the endogenous MER, lenti-blank as positive control, and non-treated cells as negative control. And we analyzed the RNA level of the genes, and as a result, as we expected, we saw that more expression in genes in lenti-off-MER condition, which was have a repression of gene expression, and on the other hand, less gene expression in MER condition, showing the real effect of microRNA. In addition, using DMI8 microscope, we captured the rate of the beating cells from each well and from each condition, and we observed different rates of beating, as you can see in figure number 4, with more palpitation and tachycardia and arrhythmia in MER condition compared to other wells. And furthermore, calcium transient tests showed more intensity of fluorescence in MER condition, showing the more calcium accumulation in cells due to dysfunctionality of the, for example, N6-1, which is the sodium-calcium exchanger, and probably other genes related to calcium handling process. To conclude, we realized that microRNAs in general, like MIR181A5P, would affect the calcium handling process, and thus these results show the potential effects and benefits of MIR on as therapeutic options in drug discovery, since there is no FDA-approved medication, unfortunately, for POTS in context of ME. Thank you very much.
Thank you so much. So much great information today. My goodness.
Thank you.
And we see lots of emojis going by, which is awesome. Does
Thank
anyone
you.
want to unmute and ask a question or write it in the chat? Okay, let's move on to the next presentation. And again, if you have a question, if you didn't get a chance to ask it, there's still going to be a few minutes, I suspect, at the end where we can come back to any of the presentations.
Sorry, I
so
do have. Oh, never mind.
Oh, it's okay. Go ahead, Michael.
So this is so much information; it's hard to take in at once. But
Yeah.
you talk about calcium transients, and I'm just wondering if there's any relation to that with the potential theory of calcium overload in the mitochondria. Is that any sort of relation to?
Yeah, exactly. So I tried to understand the expression of the genes related to three more important calcium handling gene, which has the important role in calcium handling gene in general in cells. So one of them is related to sarcoplasmic reticulum. Another one is related to remove the calcium from cytosol. So yes, generally one of them, ATP2A2, is related to mitochondria also. But the most important one was N6-1 for me, which was the calcium sodium exchanger on the membrane of the cells in order to see the effect of microRNA.
Sorry, that's some really exciting stuff. So thank you for your work.
Thank you.
Thank you, Michael. Okay, and we have our last presentation, which is reminder. And then we will have a few minutes at the end, I think, to ask some extra questions. So you guys can type them anytime in the chat, or again, you can raise your hand and unmute when the time comes. We can see your next slide. So it's small, like it's making the slides a little bit smaller.
Yeah, I'm just working that out. Sorry, I have two displays. I'm gonna swap my displays. Okay, is that much better?
That's much better. Thank you so much.
Sorry, one sec. Hi everyone, my name is Raminda. Sorry for the delay. I work with, I'm from Simon Fraser University, and I work with Fraser Health Authorities over at Surrey, BC. So I believe I'm 2 hours behind most of you guys. So my research entails working with ME patients and designing a multi-model MRI study to investigate ME. So we worked with patient partners to come up with a design which we can look at multiple different things, since ME is not a heterogeneous disorder, and different individuals experience different symptoms, and it's not always something that is directly investigated on MRI scans clinically. So we thought that maybe we could, look for some patterns that can be investigated. Maybe there are specific characteristics that we can look at that may help us come up with, along with other studies, some patterns, some profiles that we can use
So we kind of kickstarted this project, firstly to see whether we can achieve this multimodal imaging, and then we can look at some distinct patterns. So I have a screen of our methodology here, which kind of breaks down what we did. So this, let me use a pointer, that might make things a little easier. Are you able to see my screen still?
We're still looking at your first slide.
Yeah, okay.
Yeah, okay.
Yes, so yes, the participants went through enrollment through our ICanCME clinic, where we have Dr. Nakul, who diagnoses everyone who comes in and helps them manage their symptoms and disease throughout the course of their life. Before the participants go into the scan, there are some, safety screening things that we get them to do. Once they're in the scanner, they're in there for about 40 to 50 minutes, and we take some really nice scans of 3 places to measure metabolites, and these 3 places were chosen because they correlate with fatigue, pain, and sleep. So these places would be your ACC, your anterior cingulate gyrus, your brainstem, and your left dorsolateral prefrontal cortex. Since the scan takes time, we were limited to these voxels. We chose to do the single voxels so we can focus on specific regions of the brain instead of the whole brain, because then we can pinpoint what's happening in individual regions. We then went on to do some fMRI, which is functional scans, to look at their activation patterns whilst the patient participants are doing the scans. Over here at on C, we can see the locations of the voxels. So they go with ACC, brainstem, and left dorsolateral prefrontal cortex at the last 3 slides. D just goes into how we design the task. So the 2 main findings from the study, other than that, the ME participants were really happy that we're doing something that's looking more into their brain. And the project, the ME the model project is actually feasible, so participants are able to sit through it and we can get some nice results. So the first thing that we point over onto my right is the we looked at the MRS voxels, so from the 3 regions. We found a significant difference in ME participants and healthy controls, and that was in the DLPFC, so the dorsolateral prefrontal cortex region, where the ME participants showed higher levels of NAA, which is N-acetyl aspartate. In terms of the functional results, similarly, the ME participants showed greater activations in the regions that we investigated for the single voxel. So they showed greater functional activation in the frontal lobe, specifically in the left dorsolateral prefrontal cortex, and the anterior cingulate gyrus, and we see more activation. We see activations on the brainstem of ME patients, and we didn't really see any activations on the healthy control, which suggests that their brains may be working harder to maintain performance. So after completing the scans, we concluded that there are brain differences. They do exist when you look at the right level. We did have a small sample, unfortunately. So our sample for ME was around 18 participants. We found out very quickly that it's difficult for some participants to sit through the scan, and also research MRI scans are generally very expensive to do, so we ran out of money pretty quickly. But we're working with an we're working with a long COVID population, and we're looking to apply for funding so we can do more of this, to see if we can actually come up with a metabolite profile rather than just looking at one specific finding, which is, the higher NNA in the DLPFC. So we're hoping to recruit more participants, get more funding. Hopefully, you know, we'll see how the regions are different and how you know, we've already seen some nicer activations from the results, but maybe there is something else that we missed, maybe with a larger population that we may be able to see. So very fresh, a very new project that's taken off over on this side of the coast. Thank you for listening. I welcome any questions.
Thank you so much. Lots of emojis. Great presentation. I remember when this was an idea, and it's really nice to see it at the place that it's at, where, you know, you guys are getting data and being able to analyze it and then move into other audiences. I think MRI studies are really exciting for patients. We're kind of wanting to move away from just surveys of everything all the time and getting more and more objective data and imaging and things that we can really kind of show the problem to people who are new. Yeah, so I think your research is really important and is going to be very helpful in that. So thank you.
Thank you.
Any questions? All the research we've heard today has been just tremendous. I mean, it's so lovely. And we've gotten some really nice comments from folks. And there's a few people who've messaged me to say, this is so great. It's so great to see another generation of scientists coming into the space. So kudos to all of you. If there's no more questions for Raminder, I might bring back all of the presenters, ask them to turn on their cameras. And we'll just see if there's any questions for anybody else, because I know that we had to move through a few of the presenters when you guys were still, and you can take down your slide if you want, Raminder, and we'll just open it up to see if there's any additional questions.
Yes, I have a question, please.
Sure, yeah. Do you want to? Yes.
So I want to ask, you know, for the result of NAA has been adjusted for the potential confounding, the potential, you know, confounding factor like age, sex, or total brain value?
Are you asking if I didn't hear the first
last presenter? I'm asking her, you know, concerning the data, you know, for the last poster.
Do you
for the
mean
last one? Okay.
Yes, the last one for you. Yes, I'm asking about the data of NAA has been adjusted for the age, sex, or total brain volume.
Yes. So when we published our data recently, we correlated age and sex differences as well. We took that into account. So yeah, to answer your question very briefly, yes, we did correct for that.
Okay, thank you.
Yes.
That's a great question.
Thank you.
Anyone else want to ask a question or give feedback? That's okay too, if you guys have feedback for the presenters.
I unfortunately missed the first two presenters. I had a hard time logging in, unfortunately. But I wanted to ask a question for Braden. In your study, were you finding cell tissue death as well as the mitochondria damage?
Yeah, so that's something that we're actually quantifying right now. Our previous work with the long COVID patients did look into that more heavily. So we're just like one step behind with the ME cohort at the minute. In terms of, yeah, relative amounts of cell death, it doesn't seem much different thus far to the long COVID patients, but it's still very preliminary, so we can't say so much there. We are also looking into regenerative capacity within the muscle, so looking at things like presence of muscle stem cells and whatnot that could contribute to presence of cell death or inability to just recover from exercise, essentially.
Right. There's definitely a significant inability to recover, that's for sure.
Yeah, so we're actually really excited about that data coming up, and I'm hoping by this time next year that we'll have some cool new stuff for everybody to hear about.
Yeah, no, I'm really excited all you guys decided to pursue this, so thank you.
Thanks.
And just a reminder that ICanCME funds these individuals with a Stars of Tomorrow grant. So we do a round of those every year. Exactly for this reason, because we need to support those who want to come into the space, and we hope to be able to continue to support them in different ways and continue to build opportunities because we want to keep them now. So I see there's a question here. Just one sec. So I'll translate. It's a French question. For all the researchers, is there a significant difference between results observed between men or women in your studies?
We actually looked for this, so I found that there were no differences. Oh, actually, what am I talking about? Most of my sample was on female participants, but we are looking into long COVID, and that's a mixed batch. Long COVID, we're seeing sort of like 50, 40, 50, 60, around that ratio. But for ME, most of the participants that came in were female.
Okay, great.
If I can bounce on that, well, for me, it's the opposite. So for our long COVID cohort, I didn't check because we had a very, very small number of men participating. So statistically, it doesn't make any sense to compare. For ME, I haven't done it though. If we have a bigger amount of men, it's in my to-do list. So it's something I will do shortly for sure.
Okay. Anyone else?
For us, we have a 50-50 split of males and females, and we don't see any sex effects or age effects. So.
She says thank you. Would anyone else like to answer that before we potentially move on? Just for everybody listening, but also for you guys to know, today the topic of conversation at the theme of the conference day is all focused on comorbidities and their impact on research and their impact on care. We feel like this is one of the major mistakes that happens in our research space, is that people don't understand how those things come together and how they influence one another. And because we're not tracking them and we're not measuring them, and sometimes we don't even have great tools to measure them, where we end up is sometimes conflating that. For example, it could look like a medication doesn't work for ME patients when in reality, it may be a subset that it doesn't work for. And if we were tracking the comorbidities, maybe we could find which subset it did work for. So it's stuff like that that we're hoping to sort of generate dialogue about today. So I would encourage you guys to, if you can make it, do. And if not, the recordings will be provided. Whether people have signed up for the conference or not, the recordings will be provided. And there'll be a lot of, I think, great information there for you guys. Because even for senior researchers, I think this is a topic that's just starting to really be discussed. So it's really, I think it's going to be a really great day. So if you can, please do join. And if you can't, we'll make sure we send out the recordings. But I want to thank you all for your research, for coming and presenting today, for changing the format at the last minute with us and being so flexible. Everybody did such a phenomenal job. And thank you to everybody who attended and asked questions. Oh, Pietro has one final question he would like to ask. So if you want to unmute, we have time for one more.
No, sorry, it was a mistake, but I can say that it's amazing the proof that is found by the science, and on the ground with doctors, we can heard it's in your head, but in science there are a lot of proof. That's amazing, the difference between these two fields.
I know, it's amazing to come to research conferences and be able to see the great work that's happening. And then it's incredibly frustrating to then go into your doctor's office and see how none of it's translating. They're not aware. But it does take a long time to translate this knowledge. I was struck yesterday during Amy Prohl's presentation when she put up a slide of a news article that was 1956, where they were talking about ME and the connection to infections. And all I thought was, my parents were like a few years old. Like I wasn't born yet. Why are we still here? You know, there's no reason for it. So I think we just all need to keep working together and keep pushing in the right direction and keep fighting for funds so that we can fund great researchers like you. and we'll get there. We just have to keep working at it. So thank you guys all very much for coming today and for doing this. And I hope you can take part in the conference. And yeah, thank you to everybody participating. The link from yesterday will work for the conference, hopefully in a half hour. So we'll see you then. Bye, everyone.
Bye.